This is a production of Cornell University.
Okay, good afternoon, everybody. And my name is Mirza Arsyad. I'm second year's PhD student in Unique Lab. And today I'd like to present what we have been doing and also preliminary result on winter oats and pea intercropping. And firstly, I'd like to define what intercropping system is. And so intercropping or mixed cropping is simultaneous cultivation of two or more crops in the same field, and it's an ancient agricultural practice that has been implemented in for years in particularly Africa and also Asia. But recently it's just been gaining renewed interest provides more efficiency and also sustainability in agriculture. But other than that, there's also a couple benefits for in intercropping, which are increased total yield and yield stability, improve soil health, reduce inputs, and also increase resilience of the crops against pests and disease. And there's one combination that have been planted in this system, which is a combination winter oats and also peas that's believed to provide synergic complementary. That's where oats provides structural support to the peas, and pea help oats to nitrogen fixing that support the growth of the oats. And with these backgrounds, we came up with one research question, which is, how do we select the best winter oats accession for winter oats and pea intercropping system? And for answering this question, we have been doing a trials which is a winter oats and peas trial. And in our first year's trial in 2023 to 2024, we planted 94 winter oats accession from 3 different sources in combination with one winter peas variety, which was Blaze. And we planted these crops in complete block designs where monocrop and also intercrops plots planted side by sides, and the total plots was 378 plots. And for preliminary result of this trial, we use a mixed model to calculate this heritability of 5 traits. And here you can see that there's variation, or there were variation in heritability from low to high. And especially here we can see that high heritability on oats freeze damage. And then we make a box plot for oats and pea grain yield, and you can see here there's no much difference on oats grain yield if we want to compare between intercrop and also monocrop. In contrast to oats, for pea grain yield, there's not significant, but there's lower grain yield in intercrop compared to monocrop. And we tried to confirm these results by calculate the correlation between the traits, and it shows here that there's negative moderate correlation between oats grain yield versus pea grain yield. And next analysis, what we have that. We tried to figure out how the general mixing ability with this oats extraction, and let me briefly explain about general mixing ability here. So here, general mixing ability shows that how the oats extraction performs in the intercropping with pea. And for this general mixing ability, it's divided into two, which are producer effect and also associate effect. For associate effect means that how these winter oats extractions influence the oats grain yield. And for associate effect here, that represents how the oats extraction here affect the grain yield of the pea. And in this graph, you can see there's a couple green points here that represents or shows the extraction with positive producer effect and also positive associate effect. And if you want, and in detail, you can see all these extraction on the table at the right side. There are 18 extraction that have positive in both effects. However, there's also positive general mixing ability on the red point here that's located above these diagonal lines or identity lines. And then finally, after using index selection, we were able to select 72 winter oats extraction from our first year's trial and added more 16 extraction from green, which are assumed to be pure winter oats extraction, and we combine all these extraction and we plant it in our second year's trial. And what comes next is we are expecting to collect more data in this spring and also harvest the trial in summer. And I'd like to thanks for my PA, Jean-Luc Yannick, for giving me this opportunity to join this project, and to Peter, Leah, and also Tesfahun for helping me throughout this project, to Mark Sorrell and also David for providing us with some resources, and to all JS group members for constructive feedback and also great help in the field last summer, to Adrian, undergrad, who helps us during summer, and to Laurent, who provided us with plant materials and also some genotype data. And I'd also want to thank Fulbright for funding my doctoral program. And thank you, and I'm happy to take any question.
You expected that the winter pea would be more sensitive to the intercropping, and what might be driving that?
So we are expecting that to find accession that not too sensitive to intercropping. It means that it means that there's no much difference between monocrop and intercrop, and so we don't need to like establish a new breeding program, because we can use the monocrop scheme to for intercropping.
So I was wondering, you had the pictures of like the peas physically growing on the oat stalks.
Yes.
In the intercropping system. I was wondering, for that, is there any kind of difficulty that would come across whenever you go to harvest that material?
Yes. So here, yes, actually there's some challenges in harvesting this because um, the pea tend to load. So we have to make sure that it's like straight up before harvesting. So it's like, yeah, there's couple strategy before harvesting.
Okay, so today I'll be talking about doing QTL discovery in OP pollinated populations of apples. And like Charlie said, I'm in the labs of Dr. Awais Khan and Kelly Robbins. Oh boy. Okay. So a little bit of an overview of this presentation. I'm going to talk about the motivation, why do this in OP population and not just traditional biparental populations. I'll also go over a framework I'm using for simulations to look at the efficacy of QTL mapping in OP populations of apple. I also do need to mention the role of imputation and quality control, as that is starting to be is apparent as being very important in getting accurate QTL results in our market trade associations. I'll go over the results of our mapping power, our false discovery rate, and our fine mapping resolution. And then I'll end with some takeaways. The first, why open pollinated mapping in apples? So logistically, it is difficult to make biparental populations. It requires obviously crossing in the field. And with apples, there's quite a bit of extra kind of logistical things to keep in mind, like flowering time, number of flowers on the plant, the amount of pollen that you can get. So it's not uncommon to have to cross with multiple different trees and potentially even multiple different genotypes to get the amount of seed required to produce an F1 population. So with an OP population, you would just need to select a maternal plant, gather seed from that plant, and then plant out your F1 population. Additionally, there's long generation times that limit mapping population turnover. So this would go in tandem with an OP mapping scheme where you could screen for a trait early on and not have to grow it out to a full apple tree. So traits maybe like fruit quality wouldn't be the best for this scheme because you would need to spend the 5 or 6 years cultivating the population to screen for those traits. But something like disease resistance or anthocyanin content or something like that would be ideal. Additionally, research orchards provide a diverse and already established source of germplasm. So apples are highly heterozygous and don't grow true to type from their seed. So they must be maintained in an orchard as that specific genotype. So at Cornell Agritech, most of the research orchards have a couple hundred, up to 100 individual genotypes actively growing in the orchard. And then there's also the germplasm repository, which has 5,000 plus apple trees that are already established. So being able to utilize all of this diversity all at once would give you a lot broader range of genetics to pull from. And then OP populations capture natural recombination. They utilize this diversity. And if you do detect a QTL, you can be more sure that it's going to be stable across a broader range of genetic backgrounds. You're not just using a biparental population. So for my research, I'm using a population that was developed at the germplasm repository, which is 7 full SIB populations. But the one thing that these populations have in common is the maternal parent. which is what is crucial for this OP mapping scheme. So we have 7 Malus sieversii pollen parents, pollen donors, and 1 common seed parent of Royal Gala, which make 7 distinct full SIB populations, but in aggregate is a half SIB population. And with OP mapping, if you do collect from one tree, it is in an essence just one half SIB population. So this is a good proxy for studying how to do QTL mapping in an OP population. Additionally, because we know the pollen parents, it allows us to verify our results and kind of go further into the analysis in how much does knowing the pollen parent affect our ability to map. So I am using simulations to get these results. And to do that, what I'm using is that same biparental data set of the 7 full SIB populations. I calculated a genomic relationship matrix and set a QTL at a SNP that's segregating solely from the maternal parent. So that I know that it's segregating within the F1 population. I then simulate the phenotypes in that F1 population. And then I mask the SNP by removing it from the data set. I then use my model and reduce the population size 15 times to look at how effective this OP mapping scheme would be across population sizes. So I do that 15 times. I then get marker trait associations for these 15 different population sizes, and then repeat this process 40 times by resimulating phenotypes, all on the same locus. Additionally, there are different simulation parameters that I'm looking at. So there's 2 different genetic variances of 0.8 and 0.5, with the background variance being also 0.2 and 0.5 respectively. I'm looking at the same heritability for each scenario, which is 0.5, but I'm looking on 2 different linkage groups with 2 different LD levels, so kind of separate recombination rates. So one with a higher and one with a lower recombination rate to see how that affects the efficacy of the mapping in this F1 population. And then the important detail about knowing the parents is that I can then go back and compare the marker trait associations to the known QTL position and calculate power, fine mapping resolution, and false discovery rate across these different population sizes. Another thing we're finding out is that imputation is very important in OP mapping, especially in Apple. So GBS data can contain a high level of missingness. Imputation errors can lead to erroneous associations. High quality imputation improves detection of QTL, I think especially both in apples and this OP data set. And it's important for highly heterozygous individuals. You don't want to be having a bunch of erroneous homozygous calls when, say, your QTL is on a heterozygous site. So what I've done is I've detected and filtered impossible genotypes that are found in this biparental data set. So as you can see here, there are the 8 parental genotypes, the 7 sieversii, and then at the bottom is the Royal Gala. The Royal Gala is heterozygous at this particular locus, but all of the sieversii pollen donors are homozygous. And so that would guarantee that the QTL is segregating from the Royal Gala seed parent. However, in the F1s, you see some homozygous sites that have the allele that is only present in the heterozygous pollen donor. So that's likely due to genotyping errors. So I must remove those sites and then impute those and keep checking back to see if it's imputing them as being a site that's possible in this scheme. So I set those sites to missing, impute using Beagle. I'm using Beagle to be agnostic to parentage. And then this is also critical for ensuring the reliability of my mapping results, because if you do have a site segregating from this particular allele and it's overrepresented in the population, your QTL mapping is not going to be as accurate as it could be. So the results I'm getting show that power improves with QTL strength and population size. Here, there are blue and red lines indicating the recombination rate. So rather, depending on which linkage group it is, what the LD level is. So a higher recombination rate is in red, a lower is in blue. And then the effect size of the QTL is indicated by either a solid or dashed line. So the solid line would be a major trait with a genetic variation explained of 0.8, and with dashed, it's a genetic variation explained of 0.5. And what we can see here is that higher genetic variance means that you need a lower population size to get a higher power, and the threshold I'm setting is 0.8. And it seems to kind of be the case for both high and low recombination rates that you do get a higher power with a higher genetic variance explained. And again, larger population sizes and stronger QTL reduce false positives. So in this case, the lower recombination rate reduces the false positive rate with increasing population size. And everything does seem to kind of converge around around 500 individuals. So for this and the last one, I used a 2 megabase pair window around the causative SNP to define what a true positive would be. And the OP mapping maintains acceptable FDR with the proper design and proper population size. And then also resolution to the QTL improves the genetic variance and population size. So I measured the distance to the furthest significant SNP from the causal QTL across all the simulations. So each of these data points represents the average of all of those 40 repetitions that I did. And there's 15 data points per scenario for the 15 different population sizes. And with this, it all converges around, I'd say, 500 to 600 individuals with a resolution between 1 and 2 megabase pairs. So again, a higher genetic variance and larger populations equals a tighter QTL region. So some of the takeaways from this analysis is that OP mapping is effective with sufficient sample size and marker density. Power and resolution increase with QTL effect and population size, and imputation and genotype quality control are essential. However, there are some caveats to this. Because I knew the parents, I was able to see in the F1 population that there were impossible genotypes. If I didn't know the parents, I wouldn't be able to do that. So we're looking at new methods to do this kind of imputation without knowing the parents, which could include like haplotype tracking or something like that. And this has the potential for a scalable framework for perennial crop genetics. So you'd be able to collect a massive amount of seed and produce a large F1 population without having to do any crosses or go out in the field and, track flowering rate or flowering time, look at how many flowers there are per tree, collect and store pollen, make sure that there's overlap between when you collect the pollen, when you can pollinate. I'd like to acknowledge my committee, both of my labs, the Kahn and Robbins lab, Cornell Agritech, the USDA Germplasm Repository, and also the NSF NRT Fellowship. And I will open up for questions. Yes, Saren.
To what extent does flowering time, like differences in flowering time, limit the diversity of the pollen parents that end up getting pollinating in your diverse population?
So I'm not entirely sure in total, right? Because we're dealing with so many genotypes. But what it does mean is that we have to go out every year and actually track when the flowers are coming in, because it's not on the same day every year. There's also a lot of environmental variation that can go into when a plant might, or when a tree might flower. So it would completely eliminate the need for doing that. Oh,
Oh, I can read it if you want.
yeah.
From Jackson. Have you ever dealt with an impossible genotype in your F1 population that was caused by your parental genotype reference being incorrect?
Can you repeat that question?
Have you ever dealt with an impossible genotype in your F1 population that was caused by your parental genotype reference being incorrect?
Oh, I don't know. Yeah, I'm not sure. That is something I should look into because, right, if the maternal heterozygous genotype is called incorrectly, then at least with this simulation, that throws everything off. Yeah. Yes, John Lewis.
So just make sure you, thanks for the presentation, by the way. You described the considerable benefits of OP as opposed to making full sieves, right? Because making full sieves is just a lot of work, sounds like.
Yeah.
But the actual data set that you have, somebody did that work
Yeah.
7 times, right?
Yes.
So this is a lot of work went into this data set.
Yeah.
Cool. Just wanted to make sure.
Not sure if you know the answer to this. I don't even know. But for your germplasm, I'm assuming they're very diverged, like lineage-wise. So do you think there are any weird GBS calls due to perhaps chromosomal differences? I don't know how, you know what I'm trying to say, I guess.
Kind of. I mean, so the GBS was done at around, I think, between 4 and 6x depth for each of the Seversiae populations. The parents were done at a higher depth. I don't think that would be that much of an issue in this particular case because it is coming from 7 Seversiae, which are more closely related than, say, looking at a Orientalis and a hybrid and a Domestica. So I don't think that would have caused any of these problems. I think it's likely due to the depth or coverage of the GBS data.
Thank David.
This has been a production of Cornell University, on the web at cornell.edu.